Lecture is going to be a lecture where we kind of split into several different parts. We're going to be talking about, at the very beginning, the production of sperm and egg, which precedes the making of babies. As you guys can see here, we've got a picture of fetal development. We will talk a little bit about fetal development, what happens as a female progresses through her pregnancy and the development of the baby inside. That'll be towards the end of the lecture. Between talking about cell division and production of gametes and making babies and developing babies, we're going to talk about structures and stuff occurring within men, and then structures and stuff occurring in women. So that's going to be kind of the outlook for this lecture. Let's first talk about reproductive cell division. We've actually talked about cell division before in this class, but you have to think all the way back to the very beginning of the semester when we talked about mitosis. Mitosis, as we know, involves four phases. We a lot of times will say PMAT to remember those four phases: prophase, metaphase, anaphase, telophase. Here we're talking about reproductive cell division, which is a little bit different. Instead of forming, a bone cell into two bone cells, we're forming germ cells into what we call gametes. Gametes are sex cells. Egg, or eggs, in females produced by the ovaries, sperm in men produced by the testes. The whole purpose of forming those gametes is to make babies. We're all built to make babies whether we choose to or not. We're all built in order to have that occur. All organisms really are built to produce offspring. Some do it many, many different ways than we do. But it involves a haploid egg and a haploid sperm. We'll talk about what the word haploid means and what 23 represents. Those come together through a process called fertilization, and they fuse together to form a diploid zygote, and we'll talk about what diploid means. Zygote is basically just a fertilized egg, and this is how we begin our life, as a single cell, which then can go through many rounds of mitosis, developing us into full-fledged adults capable of starting the whole cycle over again. In order to form our haploid gametes, we go through a process not known as mitosis, but instead known as meiosis, or meiosis, I've heard it pronounced. This is going to form from a first cell 4 cells, because it involves 2 rounds of division. 1 cell becoming 2 cells, 2 cells becoming 4 cells. Those words that were on that previous page, diploid and haploid, are now defined. Diploid represents the number of chromosomes 46, or 23 pairs. Okay, and we abbreviate diploid as 2N. Typically, our cells are diploid. Okay, bone cells, muscle cells, nerve cells. Most of our cells are diploid. There are a few exceptions out there. But for the majority of our cells, they're diploid. Okay. Our sex cells should be haploid. Haploid is the number 23. In return, that represents the number 23. We abbreviate that with a single N. Now, 23 is exactly half of 46. Now, why are we doing that? Why are we reducing the number from 46 to 23? That way, when fertilization occurs, half of our genetics come from dad and half come from mom, and we end up as a very unique individual. Meiosis involves 2 rounds of division. There are still the same phases: prophase, metaphase, anaphase, telophase, except now you have prophase 1, prophase 2, metaphase 1, metaphase 2, so on and so forth. So we get to talk about those individually on this page. We have meiosis 1, which precedes meiosis 2. Meiosis 1 is a division beginning with a diploid cell. So diploid tells us how many chromosomes do we start with? 46, and we end up with 2 cells that are haploid, which means that there are 23. So we go from 1 2N cell to 2 cells that have the abbreviation N. During this phase, we divide or separate what we call homologous chromosomes, sometimes described as homologous pairs. We can see a set of homologous chromosomes here in the top right of our figure. Okay. Homologous chromosomes, we can see here, are colored blue and red, which helps us distinguish the fact that one of our X-looking like structures is a paternal chromosome. Here in red probably is depicting the maternal. What do I mean when I say paternal and maternal? Father for paternal, mother for maternal. Okay. Now, these are not truly X's. They're more like kind of sideways V's. The structures that make up a single X here are actually 2 different structures that are completely identical to one another. Meaning that this strand here and this strand here, let's call them strands for kind of simplicity's sake, that is completely identical to the one next to it. If you notice, there are a couple letters in here, big B, big B, big A, big A, big G, big G. Those are completely identical to one another. Notice that the maternal chromosomes are identical to themselves, but are not necessarily identical to the paternal, because mom is not genetically identical to dad, is she? And you can say it's the same thing. Dad's not genetically identical to mom. Okay. So during meiosis 1, we split those homologous chromosomes. Before we split them, something very cool happens in prophase 1. We increase genetic variability by performing what's called crossing over. And that's really what this figure at the top is showing. When those homologous pairs line up like they do during prophase 1, sometimes they get so close that they can actually exchange segments. What ends up happening is segments of these sister chromatids, or these chromosomes, I should say, not sister chromatids, separate and end up on the opposite chromosome. Okay. Now, what ends up happening is amazing, because at the end of meiosis 2, we end up with 4 different variations. Okay. Whereas if there was no crossing over, what we'd end up with is just 2 variations. 2 that would be genetically identical, and 2 that will be genetically identical. We don't want this. What we want is something like this to occur. Where each of our 4 cells is different. Now, I'm just drawing a picture to show 1 pair of homologous chromosomes. How many pairs of chromosomes do we have? 23. All 23 could cross over. Here in our picture on this page from your textbook, they're showing just 2 pairs. And I like the way that they show it is because right here in my metaphase 1, they show that this paternal chromosome is on the left, whereas this paternal chromosome is on the right. They will orient themselves very randomly. Now, we can create a hell of a lot of variation because of the crossing over that can occur with those 23 pairs, but also just the way they align themselves. Think about how 23 different pairs can align themselves, how many different ways. It's probably pretty close to infinite, right? And then you factor that in by how many different ways crossing over can occur, which demonstrates how genetically unique each individual egg and each individual sperm truly is. And that's why, unless you are an identical twin, you're going to be your own genetically unique individual. We'll talk about twins, so Sarah, get excited towards the end of the lecture. Okay. Now, the key to knowing the difference between meiosis I and meiosis II is to pay attention to if we're talking homologous chromosomes or sister chromatids. Homologous chromosomes are what we separate in meiosis I, and you see the words homologous chromosomes throughout those descriptions. You see sister chromatids throughout the description of meiosis II. You also see one becoming two cells in meiosis I. If you see the words two becoming four, it's indicative of meiosis II. The explanation and what's happening is really no different. Prophase, things are getting ready, chromosomes are formed, they're starting to move the middle. Metaphase, they're down the middle. The spindle fibers have connected, or microtubules have connected to those chromosomes or chromatids. Anaphase, they separate. Telophase, the two cells or four cells are forming. So you can kind of read through and pay attention to those processes. Hopefully it's not too difficult. Hopefully you still remember, at least a little bit anyway, of mitosis. All right, now we get to talk about men. And we'll begin talking about their primary sex organs, not the penis, but the testes, which are a pair of vulval glands where sperm are produced. The production of sperm is described as a process called spermatogenesis. The word spermatogenesis literally means sperm formation. Sperm is at the beginning of the word. Genesis, as we know, is a word that means, or suffix that means formation. And the fancy word to describe sperm are called spermatozoa, and we form them in cells, or within inside of the testes in a set of tubes called seminiferous tubules. I'll talk a little bit more about those before, or in a second. We've actually talked about the testes before. Back when we talked about the endocrine system, we talked about their role in the production of testosterone. We'll talk more about that again here today. Well, let's first talk about those seminiferous tubules where sperm are produced. The cells that actually do the sperm forming are called spermatogenic cells, which is nice. Spermatogenic cells perform spermatogenesis. We've also got Sertoli or sustentacular cells. They're supporting cells that help form what's called the blood-testis barrier, which will prevent any immune cells from attacking the developing sperm. And then not actually located within, but between the seminiferous tubules are Leydig or interstitial cells, which secrete the testosterone that we're going to talk about. Okay. Now, your male scrotum is associated with the testes. It's actually an accessory part of the reproductive system of men, but I wanted to go ahead and talk about it because they help promote spermatogenesis by raising, lowering, thickening up the walls around the testes to keep the testes a little bit cooler than 37 C, which is our typical core body temperature. We like spermatogenesis, or we prefer spermatogenesis to occur at about 34 C, so about 3 degrees C below that of our body cavity. And the two muscles that are involved with the scrotum that achieve this are the dartos muscle and the cremaster muscle. Cremaster muscle will raise and lower the scrotum closer or farther away from the body, and the dartos muscle can thicken up the wall of the scrotum to conserve heat, kind of like putting on a thicker coat, or it can relax and cause the wall to thin up, which would promote heat loss. All right, the fancy way to describe meiosis in men, as we've said already, is spermatogenesis. And as we just said, it is going to occur in those seminiferous tubules. And just like we talked about on the couple slides ago, it involves 2 rounds of meiosis, where we reduce the number of chromosomes from 46 to 23. Sperm containing 23 chromosomes are considered mature, and we'll look at how that occurs. An important distinction between men and women, and the big difference between men and women, is that spermatogenesis doesn't begin until puberty, and it lasts throughout our life. So spermatogenesis is the formation of sperm. What do you think oogenesis is the formation of? eggs. Okay? What's amazing for you ladies out there is that process actually begins before you're born. But then it basically just takes a timeout for 10, 12 years until puberty is reached. And then it starts up again. And then each month things happen in the female reproductive cycle that promote it and then start it all over again. So females are unfortunately going to be a lot more complex, and we're going to save that for Friday. We're going to have the harder part for the last part. What's also different is that men can technically still get a female pregnant throughout the entirety of our life after we've begun going through spermatogenesis during puberty. Females cannot because of menopause, and we'll talk about menopause on Friday as well. So let's talk about the steps of spermatogenesis. It begins with stem cells called spermatogonia, located in a region of the seminiferous tubule called, or near the basement membrane, which is the deeper part. And they squeeze through the blood testis barrier and then develop into primary spermatocytes. Both spermatogonia and primary spermatocytes are diploid in number. How many chromosomes does that mean they have? 46. Okay. After forming into primary spermatocytes, they then replicate their DNA and begin meiosis I. And after meiosis I, they become 2 haploid secondary spermatocytes. And that means they are how many chromosomes in number? 23. Good. Okay. Now, after forming into secondary spermatocytes, we then go through a second round of meiosis to form 2 more haploid, what we call spermatids. We can see these steps on the next page. I kind of like this image a little simpler than this one, but we have our original spermatogonium, the singular form of spermatogonia, diploid in number. Here's our primary spermatocyte developing 2N in number, 23 pairs, so 46 total. Okay. Meiosis I ends, we form the secondary spermatocytes, which we represent with the N, telling us that they are haploid in number. Now, something unique takes place when sperm development. We have these little bridges that still connect because those cells don't actually complete cytokinesis. You don't remember, cytokinesis is where the plasma membrane splits. Well, they still hang on to one another. It's like they're holding hands. Same thing happens during the second round of meiosis when we form what we call the spermatids. Okay. The purpose of this, they think anyway, is to allow for synchronized production of sperm in any given area of our seminiferous tubules. So basically, we want them to all be formed at the same time. And that way they can mature and develop at the same time. That's what I think anyway. Final stage of spermatogenesis is called spermiogenesis, because why not make it sound similar and spell similar to spermatogenesis? This is where those immature spermatids develop into the mature sperm. And you can see that there's quite a bit of difference between these cells and these guys. And so once they then develop into those mature sperm, they will then be released into the lumen of the seminiferous tubule, where they then can go fertilize the egg. The release of sperm from the Sertoli cells is a process called spermiation. So we've got kind of 3 very similarly sounding words: spermatogenesis, spermiogenesis, now spermiation. You're going to want to make sure you can tell the difference on those. Okay. Something I might also ask is not necessarily walking through the stages, but you should know which ones are diploid and haploid for sure. So, all right, structurally, sperm, pretty small. What's amazing is the amount of sperm that are produced each day. At least in my opinion, I think it's pretty amazing. About 300 million sperm go through this step, or these steps, I should say, not this one step. Okay. And if you think about it, only one is able to fertilize an egg, and how many times does that happen in a man's life? A couple, maybe none, maybe a whole bunch, depending upon the individual. So obviously, gobs and gobs and gobs of sperm are basically just going to be discarded. Okay. But structurally, like I said, they're very small, and they contain several structures that help them not only reach, but also to penetrate a secondary oocyte. Secondary oocyte is the egg that's been ovulated. We break down the structure into kind of two parts. The head is really physiologically the most important. This is where you find the nucleus, which means that's where the chromosomes are. How many numbers, or how many chromosomes? 23. Okay. Covering the end of the head is a cap-like vesicle called acrosome, which is filled with enzymes. I kind of think of it kind of like a snow plow that helps not plow through the snow, but plow through the female egg and penetrate it to allow for fertilization to occur. This process is called capacitation. We'll talk more about this on Friday, but it involves the functional changes of the sperm undergo in the female reproductive tract, allowing them to fertilize the secondary oocyte. The rest of the sperm is the tail, begins with the neck, this constricted portion where it forms the microtubules that really form everything else. The middle piece is important because that's where you find mitochondria that provide energy to help whip the principal piece and the end piece. All right. So that was the testes and sperm. What I want to talk about now are some of the secondary sex organs of the male reproductive system that aid those sperm that we just talked about. Okay. From our seminiferous tubules, sperm will be carried to the epididymis, which is kind of an extra organ on the backside of our testes, where those sperm are matured and will be stored. Okay. Within the tubes of the epididymis are smooth muscle layers that contract to force sperm into the vas deferens when a male ejaculates. So that's going to help push the sperm forward. The vas deferens, kind of the old school term, ductus deferens, kind of new school, both mean the same thing. Muscular tube that delivers sperm into what we call the seminal vesicles. The duct itself has a very narrow lumen and is well innervated by the sympathetic nervous system. These seminal vesicles are a pair of glands posterior to the base of the urinary bladder, secrete an alkaline fluid that helps neutralize acids of the vagina, fructose, which would provide energy to the sperm, prostaglandins to contribute to sperm motility and viability, and clotting proteins that cause coagulation or clotting after ejaculation. They secrete their substances into a very small set of ducts called seminal vesicle ducts. Together, the seminal vesicle ducts and the vas deferens join to form what are called the ejaculatory ducts. Okay. Ejaculatory duct passes through the prostate gland and empties into our prostatic urethra. And really, that's all the function of the ejaculatory ducts is. Your prostate gland, we mentioned the other day, surrounds the first part of the urethra, the prostatic urethra, but it is not involved really whatsoever with the urinary system. Its function is to produce some other fluid called prostatic fluid. This is a milky, slightly acidic fluid that's released into the urethra to help increase sperm mobility and viability. And then the final set of glands are called the bulbourethral or Cowper glands. Cowper's glands, they're positioned at the inner end of the penis, just below the prostate. And they function to produce a clear alkaline fluid that will lubricate the head of the penis and protect the sperm by neutralizing any residual urine. The acidity of the urine would kill or destroy or kind of disfigure the sperm, and we can't have that because disfigured sperm will not swim. They also secrete mucus to lubricate the end of the penis to decrease the number of sperm that are damaged during ejaculation. This is where that kind of pre-ejaculate, as it's sometimes called, comes from. So it's that clear fluid that sometimes starts to trickle out before the actual semen. Urethra, shared tube, as we know, between the reproductive and urinary systems. It carries urine for the urinary system. It also carries semen during ejaculation. And then the penis serves to deposit semen in the vagina during sexual activity, and we're going to talk about this. Blood vessels fill in the penis with blood, causing an erection to occur. And erections are important because they help enter the female reproductive tract. Okay. Just to kind of summarize, most of this we've already covered, but semen is a mixture of sperm and seminal fluid, typically has a volume of 2.5 to 5 mils. each of those males is going to have 50 to 150 million sperm. It's slightly alkaline pH, kind of similar to where blood is, 7.2 to 7.4 or 7.7, due to secretions from the seminal vesicles. Coagulates or clots after ejaculation due to the clotting proteins. Liquid portion of semen contains substances that help to nourish and activate sperm cells. Normal semen is only about 10% sperm, 90% other stuff. And that other stuff includes secretions from the prostate gland, makes up about a third of the total volume of semen. Contains vitamin C, several enzymes to activate sperm. Bulbourethral gland produces that pre-ejaculate, and the seminal vesicles that secrete their secretions, as we've already talked about. Okay, next couple slides I want to talk about the role in which the male brain actually regulates the production of sperm. And we describe this relationship between the brain and the testes as a brain-testicular axis. It involves three different parts: the hypothalamus in the brain, the anterior pituitary dangling from the hypothalamus, and then the testes themselves. Okay. This is what controls spermatogenesis, and it begins at puberty when cells from the hypothalamus begin secreting in large numbers gonadotropin-releasing hormone. Okay. Remember back when we talked about the endocrine system, we learned that the hypothalamus will release two sets of hormones, releasing hormones and inhibiting hormones that are carried directly to the anterior pituitary. Well, gonadotropin-releasing hormone is carried directly to the anterior pituitary, triggering the release of two hormones, FSH and LH. Okay. LH will go directly to the testes and stimulate the secretion of testosterone from those interstitial cells. We can see that here on this page. Okay. Here's a GnRH carried directly to the anterior pituitary. LH is secreted, goes to those interstitial cells, secretes testosterone. Okay. Now, if testosterone levels go up, testosterone will be part of a negative feedback loop that inhibits the release of those hormones, which would make testosterone levels continue to rise. Okay. We're going to talk about what testosterone does here in just a second. The other hormone released as a result of GnRH is FSH. And FSH acts indirectly to stimulate spermatogenesis. Okay, how it does so is by, along with testosterone acting synergistically, will act on the sustentacular or Sertoli cells to stimulate the secretion of androgen-binding protein. Okay, we can see ABP is produced by those cells. Okay. ABP will keep testosterone levels high, which allows for the final steps of spermatogenesis to occur in the seminiferous tubules. Now, another hormone, this is actually not released by the anterior pituitary because it kind of makes it seem like it is. It's released by those seminiferous tubules, the sustentacular cells, will inhibit FSH secretion by the anterior pituitary. So inhibin is a testis hormone. Another testis hormone is testosterone, and then there's also dihydrotestosterone to a much lesser extent. They're the ultimate goal of this whole axis. Testosterone promotes prenatal development, so this is even before birth, development of the reproductive system in ducts, the descent of the testes, development of external genitalia, possibly certain regions of the male brain. Development of male sex characteristics like the enlargement of the male sex organs, especially like the testes and really the penis. Development of masculine secondary sex characteristics. Deeper voice because the larynx is enlarged. The larynx is the voice box. That's why men typically have a deeper voice. Men are also much, much hairier than females typically because we get it on our chest and back and legs and arms and all sorts of other places in high quantities. So that's related to this. Development of sexual function, sexual behavior, spermatogenesis, sex drive or libido, and then stimulation of anabolism. As we know, anabolism is building up. So that's what promotes like protein building in your muscles, which is why men typically have bigger muscles and bigger bones than females. Questions on that? Okay. The figure on the right is a feedback loop describing what happens when testosterone levels go up. It involves the decreased release of testosterone, so it's negative feedback, and we talked about it over here. Okay, male sexual response begins with erection. At least typically, erection is the enlargement and stiffening of the penis, involving several vascular and neural events occurring. Now, an erection can be basically stimulated two ways. Direct contact to the penis, so, whatever that contact is, I'm not really going to get into that. But the penis, especially the glans penis, the head of the penis, as it's a lot of times known as, very sensitive because it contains several tactile receptors, which will trigger a parasympathetic reflex via afferent fibers to the brain and spinal cord. So that's what we're showing right here. Here's our mechanoreceptors stimulated by mechanical stimulation, sending in signals to the spinal cord and then also up into the brain. Okay. As a result, efferent parasympathetic fibers will send out signals that eventually cause the release of acetylcholine, VIP, nitric oxide, which all cause vasodilation of the penile arteries. Okay. This means that blood will rush into the penis. This process is called vasocongestion. Okay. As it rushes in, it fills chambers known as erectile bodies, or chambers of the erectile bodies, and a resultant pressure collapses the venous vessels, trapping blood flow. And that's where the penis will become erect and stay erect, at least for a short period of time. Okay. Now, one of the most common drugs that men take as they get older is Cialis and Viagra. Those are just basically really potent vasodilators. Okay, because a lot of the reasons men cannot get an erection as they get older is just their circulation is not as good. Okay. Now, is that the only way to induce an erection? Direct stimulation, that is, not drugs? No. Sights and smells and thoughts and sounds, those involving various emotions, involving high brain centers, can also trigger an erection. So it doesn't necessarily have to be direct contact to the penis. But emotions like anxiety and fear can also influence in a negative way the formation of erection, so they can inhibit it. Okay. Typically after erection is when emission and ejaculation occur. Emission is a release or discharge of a small volume of semen prior to ejaculation. This involves sympathetic nervous system fibers that stimulate peristalsis or rhythmic contractions and smooth muscle of the vas deferens, propelling sperm from the tail of the epididymis into the urethra. Okay. We can see that happening right here. Okay. Sympathetic signals coming down, causing release of the stuff from the epididymis. Okay. At the same time, the smooth muscle of the prostate gland will force prostatic fluid into the urethra. The seminal vesicles and bulbourethral glands will also be releasing their substances. Okay. Now, that's what we're showing here in these two images. Now, why does this occur? Typically, it's because of, eventually there's a point where there's been enough mechanical stimulation creating the ejaculation reflex. Okay. This is where, during the actual act of ejaculation, a powerful release of semen is expelled to the exterior. Okay. And this involves what we call the ejaculation reflex, which is what we're showing off over here. Okay. Sensory signals come in, efferent signals come out, stuff starts to be released. Okay. Now, does it trickle out or does it flow out in a steady stream? The answer to that is no. And that's because of the muscle that is innervated. And it's actually a skeletal muscle that's innervated, but it's really it's really involuntarily controlled. That muscle is called the bulbospongiosus muscle, kind of a cool word. It contracts in a pulsatile manner, which is why semen comes out in non-steady streams. And it comes out in kind of spurts. And then what's important here, obviously, Because the location of the ejaculation reflex centers in the lumbar portion of the spinal cord cannot be interrupted by inhibitory stimuli from the brain. And that's pretty much common sense. Once it happens, it's going to go through. Any questions on any of that? Okay, this is right where I want to stop. This is where we stopped first class. We'll pick up with the females on Friday.